All right, so we have already taken the microscope parts pre-assessment. I went back and I double-checked to make sure that I had that from everyone. It appears as if I do. There should be two documents that you have in front of you. The first one is the parts diagram. And the microscope parts diagram looks like this. We will be filling this out over the course of today. The second document that you should have in front of you is a how to use the microscope document. This gives us a step-by-step for how to appropriately use a microscope. If you could please have the first document out that has all the blanks in it, we're going to start there by filling that in. All right. Microscopes have changed drastically over time. Robert Hooke was the very first individual that we kind of recognize as using a microscope. He created his own microscope that then allowed him to identify cells. He was actually the first person to call cells cells. He thought that they looked like little rooms of a larger house. Specifically, when he first identified cells, he was looking at cork. And he noticed all these little subdivisions in cork. And he said, well, I'm going to term those cells exactly the same as monks live in rooms that they call cells. We're going to call those small subdivisions of cork cells. Microscopes have changed. Currently, the microscope that we are going to use is this microscope right here. It is a monocular, meaning one ocular lens microscope. They are compound light microscopes, meaning that they are using multiple lenses, two lenses, hence the term compound, and we are going to focus light in order to magnify. Microscopes are more complex today. We do have binocular microscopes, like the one all the way to the left, and the stage does move side to side, horizontal, left to right, by some moving devices. We don't have microscopes that are that complex. Our microscope just simply has a stage, and we move the slide around on the stage using our hand. Okay? So the microscope we will be using is similar to the second one from the left. So there is a huge variety of microscopes. There are dissecting microscopes, which we do have. The total magnification of dissecting microscopes is not nearly as high as our compound light microscopes. The dissecting microscopes are on the bottom of the cart. Please leave the dissecting microscopes on the bottom. You need a microscope that is a compound light microscope. There are also electron microscopes. Electron microscopes are the size of a room. Because we're looking at electrons, as I was to be speaking, electrons or atoms in themselves are going to wiggle or move, and the movement of atoms causes it to be very difficult to focus. So if you are using an electron microscope, you would find that the room would be padded with soundproof material so that as somebody was moving around making noise, those atoms are not vibrating to the point where you couldn't focus on them. So kind of a cool piece of equipment. We don't have the money in a public school to be able to afford electron microscopes, though if you become rich and famous and you'd like to donate one to the school, we'd love to have one. Okay? So again, Robert Hooke was the very first person to use a microscope to identify cells. We're going to walk through all of the parts of our microscope, starting with number one. Number one is the body tube. The body tube allows light to move between the ocular lenses, or the ocular lens, and the objective lens. The next structure that we need to identify is the rotating nose piece. The rotating nose piece allows us to cycle between a 4x objective lens, a 10x objective lens, and a 40x objective lens. Some microscopes have 100x objective lenses. I do not believe any of them that we have here on campus have the 100x objective lens. That is a special type of lens that we use what is called oil immersion, where we put a small drop of oil underneath the lens in order to further focus light. So 4x objective lens, 10x objective lens, 40x objective lens. Whenever we start looking at any type of specimen under the microscope, we will always start with the 4x objective lens. Do not ever start with a 10 or 40x objective lens. So the very first thing that you should do upon entering the lab today is make sure that the 4x objective lens is the one that is in the down position for viewing. The next structure, number 5 on our sheet here, is the stage clip. The stage clip is for holding your slides. In order to make an appropriate slide today, we have 2 types of slides. One is just a solid flat glass slide. The other one has a small well or divot in it. You will need to pick up that slide, and you will need to feel it to figure out what direction is the divot in, because when you first look at it, it kind of looks like a little bit of a bubble of water just on the slide. You need to figure out which direction is the well, which direction is the divot. You are going to then take and you are going to situate so that divot is facing up towards the ceiling. You're going to take a single drop of water and you're going to drop it in that divot. You're then going to take your second slide, your flat slide, you're going to lay it over top. We call this a wet mount because the water is still in between those 2 slides. We're making a sandwich of water and whatever goopy, yucky, nasty stuff you got from your water sample last week. It is in your best interest to collect some of that goopy, yucky, swampy, nasty stuff and observe that. If you got some plants in there, feel free to put a plant leaf on the slide and observe the cells in plant leaves. They tend to look very, very cool. If you're looking at like swamp water with kind of a mucky, yucky material in it, please make sure that you aren't putting too much on it, because if you put too much on it, you may find that you can't see through the slide very well and you can't really see anything other than mud. So it might require us to dilute it just a little bit. Okay? The diaphragm. The diaphragm is for allowing more or less light through the stage to your slide. This is really important because some microscopic organisms that we look at, they're going to end up with a body that's either translucent or transparent. And if we have a whole bunch of light making its way through that stage into our slide, we're going to find that we can't see some of those objects that are translucent or transparent. And we're going to really have trouble identifying things. And so reducing the overall amount of light that's moving through the slide oftentimes is important, and we do that using the diaphragm. The next one is the light source. All of our microscopes have a light source. Some microscopes use mirrors, and then it wouldn't require any electricity. And you just simply adjust the mirror towards a light source to direct light up through the stage. I believe all of our microscopes have a plug-in. If you plug in your microscope and you turn it on and the light does not come on, please let me know. That means I may need to replace a bulb. Our next structure that we need to know is the ocular lens, also known as the eyepiece. The ocular lens is the part we look through. Again, the microscopes that we use will be monocular, not binocular. We have a single ocular lens that we look through. So you'll have to situate your eye over top of the lens. You'll have to close the other eye, and you're going to focus looking down through the ocular lens. You will notice the ocular lens has a magnification of 10x. In order to determine the entire magnification, the whole magnification that we're using when we look through the microscope, we would take whatever objective lens we're using, say it's the 4x objective lens, and we multiply that by the magnification of the ocular lens. So I have a 10x ocular lens times 4x for the objective lens. What is my total magnification? 40. Yeah, 40x. My total magnification using the 4x objective lens is 40. What about the 10x objective lens? What is my total magnification? Yeah, 100, because 10 times 10 equals 100. I have 10 for the ocular lens. I have 10 for the objective lens, so my total magnification equals 100. And the same thing would occur for the objective lens. That's the 40x objective lens. It'd be 40 times 10 for the ocular lens. That gives us a total magnification of 400. Okay? The next structure is the arm. The arm is supporting the objective lenses, the ocular lens, and the rotating nose piece. This is also the part that we hold on to when we are transporting microscopes. Our hand goes on the arm. Our second hand goes underneath the base. We secure that microscope tightly to our chest. That way there's not any accidents. We don't try to hold it out away from our body. We keep one hand on the arm, the other one under the base. We keep it secured tight to our body, so when we are walking back to our seat with the microscope, there's not an accident with it. The next structure that we are going to look at is the stage. The stage is the portion where you lay your slide. It has the stage clips. The stage moves up and down in order for us to focus. In order to focus, we use two adjustment knobs, the coarse adjustment knob and the fine adjustment knob. The only time we use the coarse adjustment knob is when we are using the 4x objective lens. We never use the coarse adjustment knob when we have the 10x or the 40x objective lens in place. This is what happens when we use the coarse adjustment knob with the other two objective lenses. First, it's very hard to focus. It moves the stage up and down too quickly, and so basically what happens is you focused right through the plane of that thing that you're trying to observe, and you didn't see it. The other problem is that when that occurs, you keep trying to focus over and over and over again, and you're moving it all over. Well, eventually you're going to get to the point where that objective lens starts pushing down into your slide. There is glass that makes up the objective lens. There is glass that makes up the slide. Glass on glass does not tend to work out real well. One of the two things is going to break. Oftentimes the slide will break, and then we have to look at the objective lens to make sure it wasn't scratched. So please be very, very careful and make sure that anytime you are using the coarse adjustment knob, you are only using the 4x objective lens. When we use the fine objective lens, we have then rotated past the 4x objective lens, and we are now using the 10x or the 40x. The really cool thing about the microscope is that once I've focused in the 4x, oftentimes it is very, very, very close to being focused when I've rotated to my next objective lens. So if you've focused with the 4x and you can see all the things that you're hoping to see under the 4x, and you're like, oh, I really want to zoom in on that thing right there, you rotate to the 10x objective lens, it'll look larger, and now I can use the fine adjustment knobs very minimally in order to focus further. And the same thing will occur with the 40x objective lens. Finally, the last structure that we need to be aware of is the base. The base is the support for the entire microscope. Again, when we carry the microscope, our hand goes under the base, the other hand goes on the arm, and we secure it to our chest. It is imperative that you know the parts of the microscope. You will not be granted 2 minutes to refer to your lab notebook for the parts of the microscope. These 14 parts you need to know, because when we're in the lab and I say something to the effect of, please use the coarse adjustment knob, I would expect that your brain would then compute, oh, I know where the coarse adjustment knob is, I'm going to use it. I would not expect that you're like, oh, I need to look up in my lab notebook where the coarse adjustment knob is, then I need to go back to the microscope and make an adjustment. When I talk about parts of the microscope, the expectation is that the terms of the parts you are familiar with. All right? I hope by describing these, this is useful. I will use these terms in the lab as we work in the lab today.